Welcome to Affinity Digital again. Myself, Usha, your botany teacher. I hope you have done your exam well. That was ecology and the genetics part. So let's do the questions discussion. Okay, so this starts from ecology. Which one of the following process during decomposition is correctly described? So answer to this will be fragmentation carried out by organisms such as earthworm. This will be the correct answer. What about the other options? This should be, here it says rise to the top layer. It should not be top layer, it should go to the bottom layer. And catabolism is not the last step. Mineralization should be the last step. And humidification leads to the accumulation of a dark colored substance, humus, which undergo microbial action at a very fast rate. This should be slow rate. In humidification process, microbial action should occur at the slow rate. The answer should be 3. Match the following. Okay, here earthworm should be detritivore. A should be 2. Succession, in fact this part is deleted, but in this question this simple term has come up. Anyways, succession should be pioneer species B1. Ecosystem service should be pollination C4. Population growth should be natality D3. So A2, B1, C4, D3. Answer should be 102. Next question. Vertical distribution of different species occupying different level in a biotic community is known as. This should be stratification. Stratification means the vertical distribution of the different species component. Say suppose trees will be at the top level, then comes the shrubs at the second level, then comes the herbs that will be at the bottom level. So this vertical distribution of the species, that is called as the stratification. Which one of the following can occupy more than one trophic level in a food web? This is very simple. Sparrow. If sparrow consumes the vegetable component like seed or fruit, then it will come under primary producer. But if sparrow consumes insect, then this so not primary producer. If sparrow consumes seeds or fruit, then it will come under primary consumer. And if sparrow consumes insect, then it will come under secondary consumer. That means sparrow can be at the primary consumer level also, it can be at the secondary consumer level also. So sparrow. 3. What type of ecological pyramid would be obtained with the following data? Here this is very simple. The primary producer itself is given only 10 gram and consumer is 60 gram and the secondary consumer is 120 gram. That means obviously this has to be inverted pyramid of biomass. In a growing population of a country, if a population is growing or expanding population, obviously the pre-reproductive individuals will be maximum. Then comes the reproductive individuals, then comes the post-reproductive individual, which will have a triangular age pyramid structure or expanding population. That is why answer will be pre-reproductive individuals are more than the reproductive individuals. Growing population always will have pre-reproductive individuals maximum. 7 is not correct. This is with respect to pyramid of biomass. Energy is always upright. This option is gone. Now in this you see pyramid of biomass. Answer to this will be not correct means incorrect option is generally upright. This will be the answer. Why? Because generally pyramid of biomass in C is inverted. This is the correct option. And pyramid of numbers in the grassland is also upright. Answer should be this. This is incorrect. Biomass means let's consider phytoplankton. Phytoplankton, then zooplankton, and then small fish, then large fish. large fish. So if this we put it in an ecological pyramid graphically, in that condition phytoplankton will be very less as compared to zooplankton, because the zooplankton will consist of the exoskeletons, which will be having a more biomass content. Then comes small fish, the biomass will be more. Then obviously the large fish will have more biomass. So pyramid of biomass in the sea, this should be inverted, but here it is given upright. So this will be a answer, because here question is asked about incorrect option. Okay. Okay, next is 108. Decomposition is a process in which a detritus is degraded into a simpler substance by microbe. This statement, if we think it broadly, this is a previous year's question. Okay, sometimes we have to follow what the answers which are given by the NTA. Okay, so this statement is given correct in according to that particular year which they have given. So this statement broadly, if we consider, this is correct. Okay, but in deep detail, if we consider in respect to NCERT, then slight mistake is there. Anyways, we have to give this statement to be correct answer, because most of the place and but answer given by NTA, they have given correct this one. So this is correct. Decomposition is faster if the detritus is rich in lignin and the chitin. This one is wrong, because if the chemical component decomposing matter has lignin and a chitin, the decomposition process should be slow. So statement 1 is correct and statement 2 is incorrect. 109. Which of the following is not a method of ex situ conservation? This should be national park. These three will come under ex situ conservation, means conservation outside the natural habitat. But national park will come under in situ, conservation in its own natural habitat. So answer is 4. Habitat loss, fragmentation, overexploitation, alien species invasion and coercion. This is a very simple question. This will all are causes for biodiversity lost. Which gas produced in anaerobic sludge digester? This should be methane, hydrogen sulfide and carbon dioxide. These are all NCERT basis. Okay, microbe which act as biofertilizer are. This type of question is a little bit tricky type. We have to think it properly. Free living nitrogen fixing bacteria rhizobium. Rhizobium fixes nitrogen, but this is not free living. Rhizobium has symbiotic association with Leguminosae family. Mycorrhiza formed by algae. Mycorrhiza is symbiotic association between fungus and root of higher plants. This genus is given in the microbes in welfare part. This is actually a fungus, but here it is given algae. So this is wrong. Root nodule bacteria, Azotobacter. Azotobacter is a free living atmospheric nitrogen. It is a nitrogen fixer, but it is a nitrogen fixer. Here it is given root nodule bacteria. It is not root nodule. It is a free living. So this is also wrong. So answer to this will be free living nitrogen fixing bacteria, Azospirillum. In the microbes and welfare chapter, this genesis are given. Okay. So free living nitrogen fixing bacteria, Azospirillum is the answer. Next. Large holes in Swiss cheese are formed due to production of large amount of carbon dioxide by, this should be Propionic bacterium. Okay, next, which of the following is wrongly matched in the given table? Streptococcus, streptokinase enzyme, removal of clot from blood vessels. This is correct. We have to find out the wrong matched. Trichoderma polysporum, this belongs to family Deuteromycetes, group of fungus which is called as imperfect fungi, and cyclosporin A, immunosuppressive drug. This is also correct. It is used during the time of organ transplantation. Monascus purpureus is a form of an yeast. It produces statin. and it helps in the lowering of the blood cholesterol. This is also correct. Removal of the oil stain and the lipase. Still here, this too is correct. Okay. But Clostridium botulicum does not produce lipase. Clostridium botulicum will produce butyric acid. It will produce butyric acid. Okay. So answer to this will be this. It produces butyric acid and is used in the rating of the jute fibers. In a jute fiber removal, it is used. Sewage treatment removes suspended solid. Suspended solid is removed by primary treatment. Can be used as by control agent in treatment of plant disease. This should be trichoderma. Trichoderma again belongs to Deuteromystes. It is used for killing the pathogenic fungus which are present in the soil. Nowadays, fungicide is not used for killing fungus in the soil. Instead, a group of biocontrol agents are used so that it kills the other parasitic fungus in the soil. Which of the following is put into the anaerobic sludge digester for further treatment? Anaerobic sludge digester for further treatment should be activated sludge. Dragonflies get rid of aphids and mosquitoes. A is 2. Bacillus thuringiensis, this should be 4. B should be 4. Glomus is a fungus which has symbiotic association with root of higher plant, and fungus role is it absorbs phosphate and nitrate from the soil. So C should be 5. And baculovirus, this is used in narrow spectrum insecticidal application. D3. So answer should be C5, D3A2. This is the answer. For the commercial and industrial production of citric acid, which of the following microbe is used? Citric acid means it should be Aspergillus niger. Aspergillus niger belongs to class Ascomycetes fungus. Hemophilia never occur in women. Okay, it never occurs in women. Correct? Gene for hemophilia is located on X chromosome. Yes, this is also correct. It is an X-linked gene. But it does not explain. Okay, it does not explain. Correct? And reason is not the correct explanation of assertion. Actually, hemophilia here, for a woman to have hemophilia, both the gene, the hemophilic gene should be located on both the X chromosome. And this hemophilic gene, when they are in a homozygous condition, they are lethal. That means it leads to death of an individual. And this female, when the hemophilic gene is in the homozygous condition, when in an embryonic stage itself, inside the mother's womb only, this female fetus will automatically aborted. Why? Because the hemophilic gene, the recessive gene in its homozygous condition is a lethal gene. Means it leads to death of an individual. That is why hemophilia never occurs in case of the women. Okay. In the questions, you might get this kind of questions might be there, mother is hemophilia, but in real life, it does not exist. Because in the mother's womb itself, the hemophilic gene is lethal, so it will automatically abort it. If 8 Drosophila in a laboratory population of 80 died during a week, the death rate in the population is, this one though we will directly apply like this, initial population is 80. Okay, according to the question. And 8 Drosophila in laboratory population of 80, 8 Drosophila has died. That means this is initial population of Drosophila and this is number of Drosophila which has died. So number of Drosophila died divided by initial population of Drosophila, this will give you the death rate. So according to this 8, that means 1 by 10. That means this will be 0.1. This will be the answer. Same way we'll also calculate birth rate like this. Detritivores break down detritus into smaller particles. This process is called as fragmentation. This is a proper definition. Detritivores means earthworm. Earthworm will break down the fragmented pieces of, it can be leaves, it can be bark, it can be stem part. This portion of the dead part or detritus part is break down by the earthworm. They used to eat it, they used to consume it, and inside their body, the dead component, after digesting it, it comes out in their fecal matter. So when it comes out in the fecal matter, the product from the body, when it passed out, it is in a pulverized state, which pulverized state where it will be acted upon by the bacteria and the fungus. So the first role in decomposition is done by the earthworm, which are detritivores. That process is called as a fragmentation, and the second role will be done by the decomposers, which is actually bacteria and the fungus. So when earthworm eat the detritus part, dead part, they consume it inside the body, they digested it, and it released out from the body in a pulverized state. So that state of fecal matter is acted upon by the microorganism like fungus and bacteria, and that process will be called as the catabolism. So first will be fragmentation, second should be catabolism. So detritivore's role is the first role in the process of decomposition. Dominance. This should be, dominance means suppose one gene is there, it will have its dominant allele form and its recessive allele form. And when in a heterozygous condition, means one dominant allele, one recessive allele, the dominant allele will only be expressed. The recessive allele is never expressed. The dominant allele suppress the function of the recessive allele. So, in a heterozygous organism, both allele express themselves fully. This is not possible. Only in a heterozygous organism, only one allele expresses itself. This should be the answer for dominance. Heterozygous organism, only one allele expresses itself. Means if it is like this, in the case of dominance, only one of the allele, that means the dominant allele will be express itself. So A should be 2. After that, codominance. Codominance will be this one. In a heterozygous organism, both allele express themselves fully. C, B should be 3. Pleiotropy, that means a single gene influences many characters. C is 4. And polygenic inheritance means many gene governs a single character. D is 1. So A is 2, B is 3, A is 2, B is 3, C is A is 2, B is 3, C is 4, D is 1. A is 2, B is 3, C is 4, D is 1. Pleiotropy will be in your NCERT. It is given with respect to garden pea. One gene, SBE gene, controls the shape of the seed and the starch grain synthesis at one time. So that will be pleiotropy example. And polygenic inheritance in NCERT, it is given with respect to skin color, where the 3 pair of gene controls the human skin coloration. And in case of codominance, in codominance only, here only I'll explain the I gene, which is located on 9th number chromosome of human, has 3 allele, and that is why it is an example of multiple allele, because it has 3 allele. What are the 3 allele? IA, IB, IO. So in a population, the I gene will have 3 allele, but in one individual we will have only 2 allele out of the 3, because human is a diploid. So I gene is example of multiple allele, and out of this 3 allele, these 2 allele, IA and IB, are codominant allele. That means when they are together, IA and the IB, they will equally express. So IA will produce an A antigen, IB will produce a B antigen. So they will equally independently express. So this form of allele is called as a codominant. They are independent allele and they express themselves independently when they are together. And IO is a recessive allele. That means this 2 will be dominant over IO allele, and IO is a recessive allele. So IgN is example of multiple allele. It is example of codominant allele. It is also example of dominance. In a test cross involving F1 dihybrid flies, more parental type offspring were produced than a recombinant type offspring. This indicates that the two genes are linked and are present in the same chromosome. How will you answer this one? How will you find out whether the two genes are linked or unlinked? We will perform a test cross. When we perform a test cross, if the two genes are unlinked, then it should have 50% parental character offspring and 50% recombinant character offspring. Recombinant offspring. If the two gene is unlinked, then it will have 50% parental character offspring and 50% recombinant character offsprings if the two genes are unlinked. But if the two gene is linked, in that condition the parental character will be above 50%. Once the parental character is above 50%, then we'll know that the two genes are linked. But all the genes which are unlinked means if they are located on the same chromosome, the two gene distance will be very far. One way is that for unlinked, or the two genes will be located on the different non-homologous chromosome. So that way the two genes will be unlinked. If they are located on the same chromosome, the distance is very far, or if they are located on different non-homologous chromosome. So to find out whether the two gene is linked or unlinked, we will perform test crossed. And in the test crossed, if the two gene, if the ratio comes 50% parental character, 50% recombinant character, it is a form of two unlinked gene. But if the parental character is above 50%, that means it is a form of a linkaged. Thalassemia and sickle cell anemia are caused due to problem in globin molecule synthesis. Select the correct statement out of it. Thalassemia is due to quantitative effect and sickle cell anemia is due to qualitative effect. We have done this nicely in the class, but in this video I'll give you a short brief explanation. Now let us consider thalassemia is of two types, alpha thalassemia and beta thalassemia. Okay, so alpha thalassemia will affect alpha chain of hemoglobin and beta thalassemia will affect beta chain of hemoglobin. Let us first consider this alpha thalassemia. Alpha chain of hemoglobin is synthesized. Alpha chain of hemoglobin is synthesized by two genes, HbA1 and HbA2 gene. Okay, HbA1 and HbA2 gene, which are located on a chromosome. So suppose let us consider this HbA1, HbA2. In the NCERT, it is given HbA1, HbA2 gene. So this two gene, HbA1 and HbA2 gene, are responsible for synthesis of the alpha chain of hemoglobin. Both the genes are closely linked. They are located on 16th number chromosome. Okay, so in every human being, 16 number chromosome will be two copy homologous chromosome. So homologous chromosome, see, so two homologous chromosome, this is suppose 16 number chromosome. These two are responsible and they are closely linked. They are responsible for synthesizing the alpha chain of hemoglobin. Okay, so in all normal human being, we have these two genes located on 16 number chromosome and we have normal synthesis of alpha chain. But if out of this four genes, if one of the gene is deleted, if one gene is deleted, then what happened? The person will be a silent carrier for thalassemia, alpha thalassemia. Okay, one gene deleted. Again, if the person is deleted with two genes are deleted, then the person will have a mild anemia because now the two genes are deleted. Again, if in case a person has deletion of 1, 2, 3 gene, okay, if it is 3 gene deleted, 4 gene are responsible for alpha chain synthesis. If 3 gene are deleted, then the person will start having moderate form of anemia. When it was 2 gene, it was mild form of anemia. When it was 1 gene deleted, that was silent carrier, nothing will happen. So more the gene affected, more will be the severity of the disease. So 1 gene affected, then alpha chain synthesis will be affected. Because of that, it will be a form of silent carrier of alpha thalassemia. 2 gene deleted, then it will be a form of a mild form of anemia in alpha thalassemia. And if 3 gene deleted, then it will moderate to severe anemia will come. But if 4 gene are deleted, then the person cannot survive. It leads to hydrophytalis, in which the embryonic abortion will occur. The person cannot survive, or RBC start getting ruptured. So that will be a severe form of the alpha thalassemia if all the genes are deleted. That means in this case, more the gene is deleted, more severity will be the effect. And that's why this will come with the quantitative effect. So only 1 gene is deleted, at that time alpha chain synthesis will not be much affected. 2 genes deleted, then also mild form of anemia. Alpha chain synthesis will be less. Okay. But if more genes are deleted, there will be very less synthesis of alpha chain synthesis. That is why it will lead to all the gene deleted, means it will lead to a severe form of an alpha thalassemia. Okay. So this is a quantitative effect. 1 gene deleted, then silent carrier. 2 gene deleted, then it will be a form of a moderate anemia. 3 gene deleted, 2 gene then mild. 3 gene then it will come moderate. 4 gene then hydrophytalis, in which the person will have severe form of anemia, or RBC start getting rupture. Okay, so that means this will come under quantitative effect. But when we say with respect to sickle cell anemia, in the case of sickle cell anemia, the hemoglobin chain HbA, the gene HbA, which is located on 11 number chromosome, HbA is a dominant form of a gene, and it will synthesize beta hemoglobin chain, normal form. Okay, but if in case if this gene is mutated, the beta form of the hemoglobin chain, normal human being, all of us, we have in the 6th amino acid, glutamic acid. But in case if a point mutation occurs in that HbA gene, okay, which is located on chromosome number 11, a point mutation, which is a form of substitution, occurs, then the beta hemoglobin chain quality will be affected. Why? Because the 6th amino acid, glutamic acid, is now substituted by valine. That means normal form of the hemoglobin should be beta chain, should have 6th amino acid, glutamic acid. But due to point mutation, the quality of the beta chain will be affected because glutamic acid is substituted by valine. So sickle cell anemia is due to quality or qualitative effect, and in the case of thalassemia, it will be due to quantitative effect. More the gene affected, more will be the severity of the disease. So answer to this will be this. A disease caused by an autosomal primary non-disjunction is autosomal primary non-disjunction will be Down syndrome. Non-disjunction means what happened in the case of, it should be autosomal. Down syndrome will have 21st number chromosome trisomy. So it will have 3 copy of the 21st number chromosome. And how does this 3 copy of the 21st number chromosome occurs? This is due to the non-disjunction of the gamete of the parents. How come this a particular person is having 3 copy of the 21st number chromosome? It must be either due to the father's spermatogenesis or either due to the mother oogenesis. This I have explained in the class nicely. So if during the time of mother oogenesis in the anaphase 2, if the chromatid are not separated. 21 number chromatid, if it is not separated, or non-disjunction of 21 number chromosome occur in anaphase 2 of either spermatogenesis or either oogenesis, then it leads to this kind of autosomal disorder. Okay, so here question is asked, disease caused by an autosomal primary non-disjunction is Down syndrome. That means 3 copy of 21 number chromosome occurs in a person. Why? This is because of it can be because of father spermatogenesis, or it can be due to mother oogenesis. Mother oogenesis or father spermatogenesis. Why? Because during the time of the anaphase 2, if the 21 number chromosome, if it is not separated or non-disjunction occur, then it leads to this kind of condition. Among the following character which was not considered by Mendel in his experiments on P, it should be this, is wrongly matched. ABO blood grouping is codominance, correct. Morgan linkage, sex determination, XXXO, this is correct. Starch synthesis in P, this should be what? Pleiotropy. So answer should be wrongly matched, means this. At F2 stage in monohybrid cross, both parental traits are expressed in a proportion 3:1. F2 means 3:1. This is correct. 3 tall, 1 dwarf. The contrasting parental traits show blending at F2 stage. In Mendel, there is never blending. Blending and mixing of the allele does not occur. So assertion is true, but reason is false. Assertion is true, but reason is false. Characters represent inheritance of blood group in human. This I have already said. Human means it should be I gene. It has 3 allele. IA, IB, IO. So it will have dominance, because these two will be dominant over IO. These two will be equally codominance, and it is an example of multiple allele A, B, C. 2. A woman has an X-linked condition on one of her X chromosome. This chromosome can be inherited by, it will be both sons and daughter. Column 1 with column 2, XXXO. This should be grasshopper A3. XXXY. This will be XXXY. Female homogametic. This will be female. This will be male. So it should be 4. B4. Karyotype 45. C1 and ZWZZ. This will be in the case of bird. Female heterogametic of sex determination 2. A3, A3B4, C1D2, A3B4, C1D2, 3. Now, how does it saying female homogametic or female heterogametic here? ZWZZ means in this ZWZZ, this is female and this is male. This is sex chromosome. So how will we represent? Autosome will represent AA plus sex chromosome is ZW. Okay, AA means autosome. So what will be the gamete form for this? One will be A plus Z and the other will be A plus W. So these are 2 types of gamete which is different. So that is why we'll say heterogametic. So this is female. Female is heterogametic. This is male. Male will have autosome AA plus ZZ will be the sex chromosome. So that means one will be A plus Z. Another will also be, another gamete will also be A plus Z. So male will be homogametic. So heterogametic female, A plus Z A plus W, male, homogametic. In a marriage between male with blood group A, okay, male with blood group A and female with blood group B, the progeny had either blood group AB or B. You have to find out the possible genotype of the parent. Okay. So possible genotype means, let us consider like this. Suppose I am making this heterozygous. Case 1, heterozygous IAIO. Okay. And let's make this homozygous IB. Okay. Because it can be either heterozygous or it can be either homozygous. This is the same condition, heterozygous or homozygous condition. So IAIO, IB, heterozygous and homozygous, first case. So suppose if we cross this, then IA here, IO here, IB here, IB. So this will be AB, this will also be AB, this will be B, and this will be B. So here you see, when I make IA heterozygous, blood group A heterozygous and blood group B homozygous, at that time the offspring is coming AB or B. AB or B. So answer will be IAIO male, and IAIO male and IB female. This is the answer. Okay. But how will to remember this? Because in entrance we don't have much time for that. So you think like this: when we make this heterozygous parent IAIO at that time, and the other partner is B blood group. So both the parent is A and B. At that time, if you make heterozygous A, then the child is coming B blood group. That B blood group and AB, though it will come only because both the parents are AB. So if you make this parent heterozygous, then a child is coming B blood group and AB. AB, though it will come from both the parents. But in case if you make this one heterozygous, then if the homozygous parent IAIA and will make this B heterozygous like this. Okay. In that case, what type of blood group will come? It will come either AB or it will come AB and A will come. That means when we make heterozygous IAIO along with the AB, it will be producing B blood group. So it will be ulta. That means when we make B blood group heterozygous along with the AB, it will come A. So like this we'll remember. Okay. So answer will be 1. The number of contrasting characters studied by Mendel for his experiment, each characters that he studied, there was 7 in number. In a cross between a male and female, both heterozygous for sickle cell anemia gene, what percentage of the progeny will be disease? Heterozygous for sickle cell anemia. Sickle cell anemia is due to a recessive gene. So suppose A gene is there and normal form of gene is dominant allele, recessive is, let's consider sickle cell anemic gene, okay, which is a recessive gene. So if a question is set here, heterozygous sickle cell anemia, that means it will be like this. Will represent like this. Okay. So normal form of the person will be AA. This will be heterozygous, and the disease will be, of course, recessive sickle cell anemia. They will express when the recessive gene is in the homozygous condition. When this condition, it will express. Disease will be a small like this. So in that case, this male here, this is female here. If you cross this, this will be totally normal person. with normal hemoglobin. This will be heterozygous. It will be heterozygous, it will be carrier, and this person will be a diseased sickle cell anemic patient. So recessive homozygous condition, this will be a sickle cell anemic patient, and this too will be heterozygous, that means which is a carrier of the sickle cell anemia. Question, what is asked here? Disease. Disease means out of 4, this will be the disease. That means answer will be 25. The mechanism of DNA replication is semi-conservative in nature. This is correct. Each of the complementary strand of the parental double helix is conserved during the process. And it also explained, this should be 1. That means what? This is a 1 strand of DNA, 5 prime, 3 prime, 3 prime, 5 prime. When it will replicate, at that time it will form 2 strands like this. From this parental strand, it will form another strand. From this, it will form another daughter strand. So 2 strands will be formed. So in this 2 strand, 1 strand come from this parent. This daughter strand, also another strand come from this parent. This process is called as what? Semi-conservative. The 2 daughter strand, one of the daughter strand comes from the parent. Okay. If a colorblind female, colorblind female marries a man whose mother was also colorblind. Marries a man whose mother was also colorblind. Okay. What are the chances of progeny having colorblindness? Colorblind female marries a man, okay, whose mother was also colorblind. That means this person's mother was also colorblind. If the mother is colorblind, obviously the son has to be also colorblind, because both the X chromosomes has a colorblind gene. So obviously this will also be 100% colorblind. Progeny having colorblindness, and the female is also colorblind. This will be this. This will be this. And this one will be, this will be totally colorblind girl, colorblind boy. This will be also colorblind. This will also be colorblind boy. That means answer will be 100%. Colorblind female, okay, marries a man whose mother was also colorblind. The mother is colorblind. That means this man also has to be colorblind. 138. Okay. Recombination frequency between gene A and C is 5%. We know recombination frequency is equal to the distance between the 2 genes. This we have done very nicely in the class. Okay. Recombination frequency is equal to the distance between the 2 genes. So here it is given A and the C is 5 map unit. Recombination frequency is equal to map unit between 2 genes. A and the C is 5. B and C is 15. B and D is 9. A and B is 20. A and B is 20. A and the B is 20. C and D is 24. C and D is 24. Okay. And A and D is 29. A and D is 9. That means A, C, B, D. A, C, B, D. Recombination frequency is equal to distance between two genes. So like this we'll calculate. Euchromatin is loosely packed chromatin, correct. Heterochromatin is transcriptionally, it should be inactive. This is wrong. Histone octamer is wrapped by a negatively charged nucleosome. Histone octamer will be there. H2A, H2B, H3, H4. 2 copy will be there, and this will be wrapped by DNA. Okay. So histone octamer is wrapped by negatively charged DNA nucleosome. This is correct. Histone are rich in lysine and arginine. This is also correct. A typical nucleosome it should be 200 base pair. So A, C, D. Loosely packed euchromatin. Histone octamer is wrapped by negatively charged DNA molecule. Octamer is wrapped by negatively charged AC and D. AC and D. And D. In E. coli strand, I gene get mutated. What happened here? Suppose I gene is here, then promoter will be here, I gene will be here, then promoter will be here, operator will be here, then structural gene will be here. Z, Y, A. Okay. So question is starts here. I gene get mutated. This has undergone mutation, and its product means repressor. Okay, repressor protein cannot bind the inducer. Inducer means lactose. Okay. What if growth medium is provided with lactose? What will be the outcome? Okay. Now, actually, when in the absence of lactose, what happens when lactose is absent? When lactose is absent, at that time, the repressor protein goes and bind to the operator. Lactose absent. Repressor protein goes and bind to the operator. So, RNA polymerase cannot bind to the promoter, and so transcription of the structural gene does not occur. Lactose absent, repressor goes and bind to the operator, so it is RNA polymerase cannot bind to the promoter. That's why transcription of structural gene does not occur. That is when lactose is absent. Okay, but when lactose is present, then what happened? When lactose is present, then lactose should come. Lactose should come. It should bind to this repressor protein, removed it, and RNA polymerase should bind to the promoter, and structural gene should transcribe. Okay, when lactose is present, lactose act as an inducer. It should remove the repressor protein, so RNA polymerase bind to the promoter, and transcription should occur for structural gene. But here in this case, this repressor, it cannot bind with inducer. The repressor cannot bind with inducer. Repressor is there. It has been bound to operator. But repressor cannot bind with inducer. Actually, inducer should come, repressor should be removed. But here question is saying that repressor jo hai, it cannot bind with the inducer. So since inducer, that means lactose cannot bind to the repressor, it will not be able to remove. So since it cannot be able to remove, what happened? Transcription cannot occur. No transcription, no translation. Okay, so that is why answer will be ZYA gene will not be translated. I repeat again, this I have said many times in the class also. Since then, I'm saying it again. What happens is when lactose is absent, repressor protein should go and bind to the operator. So when it binds to the operator, RNA polymerase cannot bind to the promoter, the structural gene cannot transcribe. No lactose, no enzyme, fine. But when lactose is present, lactose should come, it should remove this repressor protein. Lactose means inducer, it should remove this inducer, it should remove this repressor protein. RNA polymerase should come, bind to the promoter, transcription should occur. But according to the question, it's saying inducer cannot bind to the repressor. Inducer cannot bind to repressor. Inducer cannot bind to the repressor. So since inducer cannot bind to the repressor, what happened? It will not be able to remove. Repressor is not removed. That means transcription will not occur. Inducer should come and remove it, but inducer cannot bind to the repressor. So it cannot bind to repressor. That means transcription, RNA polymerase cannot bind to promoter. Transcription will not occur. Okay. DNA polymerase catalyze polymerization only in one direction. Polymerization always occur in 5' to 3' direction. During replication of DNA on one strand, the replication is continuous, while on the other strand, it is discontinuous. This is correct. This should be are correct. Both statements are correct. Bacteriophage. Again, these are previous year's questions. Majority of them are previous year's questions. I am now only focused to previous year's questions only, since you are about to appear your entrance in the next 2 months. Bacteriophage 174. This should be 53862. Lambda bacteriophage will be 48,502. B1. E. coli will be C3. Oh, sorry, C4, sorry. E. coli should be 4.6 into 10 to the power 6 base pair. Human should be 3.3. E3. A2. A2, okay. 24, 2, A2. B1. A2, B1. A2, B1. C4, C4, B3. A2, B1, C4, B3. D will be 3. A molecule that can act as a genetic material must fulfill the thread given below, except this. It should be stable. Here it is written here, unstable. It should be stable. Okay. Others are all correct. During DNA replication, Okazaki fragments are used to elongate. Okazaki fragments are used to elongate this way. This will be leading strand. Five prime, 3 prime, 5 prime, 3 prime, 5 prime, 5 prime. So this will be leading strand. This will be lagging strand. What happened here? Lagging strand, Okazaki fragments are used to elongate. This should be the lagging strand away from the replication fork. Okazaki fragments has to be lagging strand only, and this will be away from the replication fork. Replication fork is here, to be away from the replication fork. And leading strand should be towards the replication fork. Okay, so the lagging strand away from the replication. This is replication fork. Lagging strand should be away from the replication fork. Lagging strand only are called as Okazaki fragments. RNA polymerase 3 will be tRNA, 5S rRNA, and snRNA. Monocistronic genes are found in prokaryote. Monocistronic means one promoter controlling one cistron or structural gene. That is called as a monocistronic. Monocistronic gene should be found in the case of eukaryote, and polycistronic should be found in the case of prokaryote. Okay, generally, not always. The term should be used generally. Monocystronic genes are generally found in eukaryote. Polycystronic are generally found in prokaryotes. Polycystronic means one promoter controlling more than one cistron. One and two are what statement 1 and 2 are incorrect. Statement 1 and 2 are incorrect. This means monocystronic gene should be found in eukaryote. Polycystronic should be in the case of prokaryote. So 1 and 2 are incorrect. Chromosome 1 has 231 gene and Y has 2968. This is wrong. This is given ulta. Chromosome 1 has 2968 gene and chromosome Y has 231 gene. Okay, this is wrong. Repetitive sequences of DNA are thought to have direct coding. This is again wrong. They are non-coding. Repetitive DNA are non-coding. Statement 1 and 2 are incorrect. Non-coding. Incorrect. Identify the correct statement. RNA polymerase bind with Rho factor to terminate the process of transcription in bacteria. This will be the answer. Split gene is not prokaryote, it should be eukaryote. Okay. It is not coding strand, it should be non-coding strand. Methyl bonds added to 3N, it should not be 3N, it should be 5N. So it should be 2. DNA polymorphism forms the basis of what genetic mapping and DNA fingerprinting. Hemophilia is a sex-linked recessive disease. This is correct. It is a sex-linked recessive disease. Down syndrome is due to aneuploidy. Like aneuploidy means loss or gain of the chromosome other than a normal chromosome number. Of course, Down syndrome is due to gain of one extra chromosome. So this is correct. Phenylketonuria is an autosomal recessive gene disorder. This is also correct. So, sex-linked disease, recessive disease, this is correct. A, B, C. A, B, and C are correct. Sickle cell anemia is not an X-link. It should be autosomal. It is autosome. It is located on chromosome number 11. Okay. Thank you then.